the collective goal of neuroscience is to understand how brain works. This is a daunting task given the extreme complexity of the brain itself, with millions to billions of neurons and a similar number of non-neuronal cells in the mammalian brains, and the intricate interconnections among all these cells. To tackle this complexity, it is necessary to take a multi-level approaches to understand organizing principles of the brain at these different levels. At the Allen Institute, our program is also structured along these lines, along these different types of approaches, with our largest effort at the first level in trying to understand the gain a deep knowledge and understanding of the brain cell type diversity and disseminate that information to the community. And with a gradually more focused approach as we move up the ladder to more complex questions and systems. So why studying cell types? I think we all agree that in order to study and understand a complex system, one essential first step is to generate a parts list of that system. And one level of parts list for the brain would be a cell type category. With that kind of a parts list in hand, then we can identify marker genes for each cell type and create a genetic access to those cell types, as you have heard from the talks yesterday, and begin to interrogate and understand the function of those cell types in a reproducible manner across different labs. With the parts list and the genetic tools in hand, we can then begin to make discoveries about new cell types and new functions, as well as to understand other important biological processes such as development and evolution, and of course, ultimately understanding the mechanisms of the diseases and finding ways to treat those brain diseases. So with all this importance, how do we study cell types? How do we generate this parts list? We believe that in order to generate a comprehensive, unbiased parts list for the brain, we need to take a systematic and quantitative approach to profile individual cells in the brain in a quantitative manner and across different modalities. For example, we want to know how cells vary in their properties at a molecular level, at physiological, morphological, connectional, and possibly functional levels, and use a data-driven approach based on those quantitative measurements to classify cells into types, and in particular, to look for congruency of the properties across the different modalities to arrive at a unified definition of cell types. So with this kind of understanding at the Allen Institute, we have established a number of technology platforms to define cell types and then use that cell type information to study the connectivity and function, in particular systems such as the mouse visual cortex. Our front-end approach is this highly scalable single-cell transcriptomics method, which we employ to profile hundreds of thousands to millions of individual cells and generate transcriptomic taxonomies to give us a first iteration of knowledge about the transcriptomic cell types. At the other end of the spectrum of our approaches, we use a number of methods to investigate the connectivity among cell types and individual cells. We do that at both brain-wide level, especially in the mouse brain, but also at a synaptic level in the mouse and human brains as well. And look at the connections in populations of cells, and in particular, again, for individual cells. We also study the in vivo properties of the different cell types in awake behaving animals, particularly in visually guided behaviors. So how do we bridge this two very different types of information? We take three major approaches to do that. The first is to generate genetic tools using the information that we have gained from the transcriptomic taxonomy to target different cell types. As David said, our initial effort was in generating a large battery of transgenic mouse lines to target cells. In recent years, we have gradually switched to generating viral vectors, taking advantage of enhancer elements identified from the molecular data to be able to more efficiently target different cell types. We also use multi-model integrative approaches such as the patch-seq to collect triple modality information from the same cell and use that information to derive integrated definition of cell types that then can serve as Rosetta Stones that bridge different levels of characterization. And then finally, we have also established a spatial transcriptomics or multiplexed FISH platform to not only use that to understand the spatial distribution and relative proportions of the different molecular types, but also to use it as a post hoc method following our in vivo or in vitro connectional or functional studies to decode the molecular identities of the cells that we have investigated in those connectional and functional studies. Importantly, this large set of efforts, platforms, and pipelines that we have built have now been integrated into the community-wide brain initiative effort. We are now a major participant in the NIH Brain Initiative Cell Census Network, in which we work together with other investigators across the country and the world, using the same set of approaches that I just mentioned to profile cells from several different mammalian species, from mouse to non-human primate to the human brain, with the goal of deriving cell type atlases at a brain-wide scale across multiple species with spatial information, multi-model integration, as well as the connection between cell types and wiring diagrams. So our effort, so more specifically, our effort started with the study of cell types in the cortex. There are two major classes of neurons in cortex. The first is the glutamatergic excitatory neurons, which are generated developmentally within the cortex per se from the ventricular subventricular zones, migrating radially upwards in an inside-out manner to form the major layers of the cortex. And the excitatory neurons are also organized based on their laminar distribution and their long-range projection patterns. The largest group, subclass of cortical excitatory neurons are the cortical projecting intrathalamus or IT neurons that are distributed across all 6 layers, from layer 2, 3 to 4 to 5 to 6. A second major subclass is the layer 5 pyramidal tract or subcerebral projection neurons that are the major output neuron types that send axon projections in many parts of the subcortical areas. The third major subclass is the cortical thalamic projection neurons that are located in layer 6 of the cortex with primary almost exclusive projection targets into the thalamus. There are also a couple of other smaller subclasses of neurons, excitatory neurons, which I will mention later in my slides. The other major class is the GABAergic inhibitory interneurons, which are highly differentiable by their electrophysiological properties, intrinsic electrophysiological properties, as well as their elaborate local axon arborizations. These neurons can also be differentiated by a set of neurochemical markers. And this developmentally, GABAergic interneurons were not developed within the cortex per se, but in the subpaleo structures, the caudal and medial ganglionic eminence, and then migrated tangentially into the cortex. So as I said, our front-end approach is to use large-scale transcriptomics to generate a first version of taxonomy. We take two approaches to do that. One is the full-length deep sequencing low-throughput SMART-seq method. The other is the droplet-based 3' sequencing multiplexed high-throughput method. In our first set of studies, published work, we profiled cells from both mouse cortex, 2 cortical areas, and a human cortex, one area, using the SMART-seq approach. In both studies, we find that the cell types in the cortex are organized in a hierarchical manner. This hierarchical structures are well conserved across species, as well as the major cell types defined as the major branches of this hierarchical structure. In addition to the conservation, we also find species differences, mainly at the terminal, the finer leaf level of the hierarchical tree, as well as at the individual gene expression level, even for the major branches. We also compared cell types across different cortical areas using this mouse study for 2 cortical regions, primary visual cortex at the back end of the cortex and anterolateral motor cortex at the front end. The major finding from that study was that we found that the GABAergic interneuron types are shared across those 2 different areas, whereas the glutamatergic neuron types are largely distinct. This is reflected not only in the distribution of the cells from those 2 different regions, but also the number of differentially expressed genes from those different regions in homologous or same types. So that's the finding. That was the finding from 2 cortical areas. What happens if we have looked at more cortical regions? Would the finding change? And also, how do we relate our finding with other cortical structures that are evolutionarily related to cortex? So in this set of study that we recently completed, we proceeded to profile all regions in the mouse isocortex or neocortex, and that includes all the sensory motor areas, as well as the prefrontal, medial, and lateral associational areas. We also profiled the entire hippocampal formation, and also called archicortex or allocortex, which is the closest relative evolutionarily to the isocortex. For hippocampal formation, we profiled not only the hippocampus itself, but also the neighboring subicular complex, as well as lateral and medial entorhinal cortex. So in this large-scale study, we used both 10x and a SMART-seq approach, totaling more than 1.2 million cells. We combined the data together, generated this very large integrated transcriptomic taxonomy. Furthermore, we used the marker genes identified from all the different clusters of this taxonomy tree and their in situ expression patterns to identify the location, the anatomical location of all the different clusters or types. And this detailed work allowed us to annotate this taxonomy tree with anatomical location, including both regional specificity and layer specificity. And that information is shown here. So do the principles we discovered earlier still apply when we expanded our work in such a larger scale, covering so many regions now and 2 major brain structures? The principles, so here I've said that the principles still apply, but also there are new extensions and discoveries. First of all, the cells still follow this kind of hierarchical organization. And we, to describe this hierarchical organization, we named the cells at the different levels. The first level is class, and then the second level is neighborhood. The third level is subclasses shown here. The fourth level is what we call supertypes, which are the major branches of this taxonomy tree. And the final level is the terminal leaves, individual clusters. For our work is mainly focused on neurons, and we have a limited profiling of non-neuronal cells. So in this talk, I will not talk about the non-neuronal cell types in detail at all. For GABAergic neurons, we found that vast majority or most of the cell types are still are shared or conserved across all different cortical areas, including neocortical, isocortical, and hippocampal formation areas. However, at the same time, as you can see here, we also identified a number of highly cell types, new cell types that are highly specific to particular regions. And all these regions you can see here are shown in this greenish color, meaning that they come from hippocampal formation. So we now, in addition to the shared GABAergic types, we also identify a set of highly specific cell types that are specific to hippocampal formation. The landscape on the glutamatergic side is much more complex. There is still a high degree of distinction between cell types, especially between isocortex and this greenish colored hippocampal formation. But at the same time, as you can notice, obviously, that there are then a lot of cell types that are also shared across different regions. So let's take a closer look of what happens. There's a lot, I should say that there's really a lot to unpack in this taxonomy tree, and it's kind of detailed in our manuscript shown here. So for this talk, in the interest of time, I only highlight a few key points that we discovered. The first is that we identify highly distinct cell types between cortex and hippocampal formation, as shown in this UMAP. The isocortical cell types are all concentrated in this several major continents. And as you can see here, the colors are mixed together. Colors representing different regions, meaning that there's actually a high degree of sharedness across different cortical regions of those major cell types. The big block is the IT neurons, as I mentioned earlier, across all different layers. There's a distinct block here that's layer 5 PT. And then there's another block here that's actually three subclasses of neurons, the layer 6 CT, layer 5 MP, and layer 6B. There's finally a distinct one, a highly distinct neocortical subclass here. We call it layer 6 CAR3. That's also related to the cortical projecting IT types. And then besides of those major continents, then we see a lot of little tiny islands, greenish islands that are cell types that come from hippocampal formation that are kind of strategically positioned right next to those major types. To investigate this further, we performed a homology search for each hippocampal cluster to find its closest neighbor in the cortex part. So basically identify its most closely related cortical subclasses. And amazingly, we find this high degree of correspondence between hippocampal cell types and cortical cell types. We found that there's a large set of IT-like neurons in the entorhinal cortex and in the para, post, and pre-subicular areas. We also find a set of cell types that are highly homologous to the layer 6 CT, NP, and layer 6B subclass of neurons, again, in entorhinal cortex and subicular complex. Most surprisingly, we found that there's even a remote but a significant homology between CA1 and subicular, majority of the CA1 and subicular cells to the layer 5 PT neurons in the cortex. This homologous relationship was further confirmed by a large set of shared marker genes, layer-specific marker genes, as well as the anatomical location of those marker genes. Namely, those marker genes are present in the same or similar layers between isocortex and hippocampus, supporting their homologous relationship of the cell types. Another major finding is that in addition to the many highly distinct cell types that we discovered, we also found large-scale gradients that are distributed in both cortex and hippocampus. First, the gradients are in multiple dimensions. First, we use the IT cells example to show the vertical gradients of the continuous transitioning of IT cells across the different layers from superficial to deep, making the border of different layers really fuzzy. We also identified horizontal gradients or continuous variation of cells across the cortical sheath. And that's the reason behind the shared cell types across neighboring or multiple cortical regions. And we find gradients in both anterior-posterior direction and medial-lateral direction. We find that the anterior-posterior direction, the gradient is more continuous, whereas the medial-lateral is more discrete. Similarly, we find multimodal gradients across hippocampus, and in particular, CA3 itself, and then the combination of CA1 and cerebellum. Multi-dimensional gradients have already been identified in previous studies. However, due to the different natures, different methodologies employed in different studies, the exact nature of the cell type distribution across the different gradients in hippocampus and cerebellum has not been clear, and there has been quite a bit of debate in the field about this. So our transcriptomic taxonomy cell type data now is able to integrate all this information together. With one shot, we identify the gradient distributions of cells and their gene expression patterns collectively together. So in this diagram, we showed proximal to distal gradients in this direction, dorsal ventral gradient in this direction, in a vertical direction, superficial versus deep layer distribution within hippocampus, CA1, CA2, and CA3, is shown by this grayscale, and the activity level as indicated by immediate early gene expression shown in color. And providing a systematic comprehensive view with each gradient distribution mediated by a different set of genes. I think this demonstrates the necessity and power of using this kind of systematic approach to resolve the biological complexity. So as a summary for this large-scale study, we illustrated all the different cell types that we identified in cortex and hippocampus in this one slide together. This is illustrated at the supertype level. I think this diagram demonstrates several major principles, organizing principles we discovered in those two different areas. First, in the isocortex, we see large-scale shared cell types with gradient distributions across the different cortical regions. We also identify a number of highly specific cell types specific for certain regions. In particular, we find that retrosplenial cortex and anterior cingulate cortex are most unique among all the cortical regions. They have a number of cell types that are unique to themselves. The combination of this kind of a unique cell types and gradient distributions of the shared cell types collectively define the cortical modularity, and that can correlate with the modularity that's identified, by connectional studies and brain imaging studies, and could, explain that modularity from the molecular base. The comparison between isocortex and hippocampal formation demonstrates a parallel organization between the two different structures with superficial IT cells coming from entorhinal cortex and parallel post pre-subicular regions, as well as the deep output, the corticothalamic output cells in those regions. And we identified CA1 and subiculum cells as the layer 5 PT-like major output structures that send output connections to many other cortical and subcortical regions. And this molecular structure is actually that we identified purely from the transcriptomics data is highly consistent with the existing known inter-areal connections of the hippocampal formation itself. So that suggests to us that we may be able to find the molecular basis for that connectivity, identify specific cell types that are mediating those connections, and furthermore, using our molecular information to further derive the wiring diagrams in much greater detail for those structures. Okay, so with that, I'll switch to the other studies in much less detail now. The second major study, 5 minutes, yeah. I will try to summarize the other studies quickly. We use this patch-seq approach to identify cell types in an integrated manner. We have done this using the mouse visual cortex GABAergic interneurons as an example. We profiled more than 4,000 cells and obtained electrophysiological and morphological, and transcriptomic information from all those cells, as well as morphological information from 500 of those 4,000 cells. We mapped every cell into our transcriptomic taxonomy tree, and we find that amazingly those even GABAergic inhibitory neuron types are also distributed very often in a layer selective manner, and there's often a gradual or continuous transitioning across layers for those transcriptomic types, even for those interneurons. We further can use electrophysiological information to classify the cells and use morphological information together with the EFIS properties to independently derive a set of ME types. To integrate transcriptomic information together with the ME information, we generated this cross-correlation plot for the exact same set of neurons. And we see that the T types, the transcriptomic types, are highly correspondent with the ME types at a high level, as in this diagonal distribution of the cells. At the same time, we also notice that this correspondence is not perfect. It's not one-to-one. There's often this kind of fuzziness at a fine-grained terminal leaf level for both types of classification. Then we use the mapping statistics to integrate the ME and T information together to define 28 what we call MET types. With this kind of definition, the MET types have consistent marker gene expression and morphoelectrical properties that are actually highly mutually predictive. This mutual predictivity is actually critical for us now to link disparate data sets and investigations together. So that's for GABAergic interneurons. For long-range projection neurons, we cannot study them just by patch-seq because the long-range projections are lost. So we resorted to this full morphology reconstruction approach to look at the projection patterns and try to correlate that with their molecular identities. And here, I want to show 3 examples very quickly to demonstrate actually the big surprise that we have seen. The first is the layer 2, 3, 4 IT neurons, intracortical projecting neurons. And we find that amazingly, every cell, their cortical projections, every cell is different. And this is the projection map. Each neuron picks a subset of the projection targets of that particular class of neurons, and this subset appears to be random so far, and they do not correlate with our transcriptomic types, which are arranged across according to cortical depth, because there's no correlation between the projection patterns and cortical depth, as we have found so far. The second is the layer 5 PT, the long-range projection neurons. And here, again, we see a tremendous diversity of long-range projection patterns at the individual cell level across multiple cortical areas. In some cases, there's excellent correspondence between projection and transcriptomic types, as shown in our previous studies. The medulla projecting and non-medulla projecting cells clearly belong to two different transcriptomic types. However, beyond that, the projection diversity cannot be explained by transcriptomic classification yet. And finally, the most amazing example is this layer 6 CAR3 subclass of neurons, which is shared between the layer 6 cells from lateral cortex and the claustrum. This is a highly homogeneous transcriptomic cluster. However, each individual cell belonging to this subclass from either claustrum or cortex exhibits extensive, elaborate intercortical projections that are different. Every cell is different and unique. And we found that the projection patterns follow this topographical rule from the anterior to posterior, rather than following the transcriptomic identities. So with that, I need to close my talk. Now, this is the final thing I wanted to say, is that we also performed the correspondence studies between single cell transcriptomic and single cell epigenomic data. And this is in collaboration with my colleagues at the BICCN. We are able to integrate multiple single cell data sets together. And importantly, such integration then allows us to associate chromatin cis-regulatory elements with specific marker genes in a cell type specific manner. And then that enables us to identify cis-regulatory elements, which we can then use to generate viral vectors to achieve cell type specific targeting. The story is, of course, is more complex than what than you can imagine here. The success rate is not 100%, but we were amazed to see that a significant proportions of enhancers we tested can target specific cell types, especially at the subclass level, in a quite a significant, specific manner in both mouse, and now we're doing that in the primate brains as well. So with that, I want to summarize what I have told you. I showed you that large-scale single-cell transcriptomics provides a framework for cell type classification. We found that different cortical regions have shared interneuron types, but we also identified a set of unique cell types for hippocampal formation in addition to the shared ones. We find that excitatory projection neurons are highly distinct between regions. They also exhibit gradient distributions and gradual transition and homology between neocortex and hippocampus. Integrated classification allows us to identify MET types that have congruent properties and high degree of mutual predictability. On the other hand, long-range projection neurons exhibit extensive projection diversity even in relatively homogeneous transcriptomic types. Combination of transcriptomic and epigenomic profiling enables enhanced discovery and the development of novel cell type targeting tools. So in summary, I wanted to convey the message that all these different multi-model characterization of cell types are now really coming together to give us this comprehensive picture about cell types, which will allow us to bridge analysis across different modalities and across species. With that, I want to thank a large team of people at the Allen Institute that contribute to this tremendous effort. Especially want to thank our founder, Paul G. Allen, for his vision, encouragement, and support, as well as our funding agencies at NIH. With that, thanks everyone for your attention. I'll stop sharing now.